don't forget. Again, I apologize for some of the in and out of the connection. That I'm sure has a lot to do with the infrastructure being put back together after the hurricane. If we do lose connection, as I told you at the beginning of the semester, I will, either you lose connection on your end or my end or however that happens, I will continue the lecture and simply post the recording later on, which you can then listen to when perhaps connections are a little bit better or whatever the case may be. How's everybody as far as connection right now? I'm going to leave the video off because it seems like every time I turned it on, it's stressed the connection and actually cut me off anyway. So, and seeing me first thing in the morning may not be the most exciting thing in the world to do anyway. Audio's good, very good. That's the most important part. All right, very good. Any questions for me? Any concerns? Remember, we do have our exam on Tuesday. It will be in the lab. It will be done with online, but with bringing laptops. And then after the after lecture, we will then of course have lab. We'll be doing lab number 4, which I posted this morning. There's 2 parts to it. Please make sure that you download electronically or otherwise both of the parts. Okay. No questions. We're good. All right, fantastic. So what we were doing was we were talking about cells, and the first couple of things we went over were just some pretty obvious things. You know, we talked about, you know, cell division and the importance of that, and we'll come back to that later on in the semester when we talk about particularly how it happens. Now, you might remember from, I don't know, maybe high school biology that there are really kind of 2 ways that cells divide. So it's a very fascinating topic, but we'll return to that a little later on when we get to it. The first thing we got to know is all living things are made up of cells. Very nice inductive statement on all that stuff we talked about in the first week. Inductive statements, again, are those sort of generalized statements. Like I asked in class, have we seen all living things on Earth? No, without a doubt we haven't. Every so often somebody reports a new insect or new worm or new fish. Rarely a new mammal. Rarely a new big thing, you know what I mean? We've seen most of the big things. Now, and that may be different in the ocean, but even if we haven't seen it, we know that as far as we know, life needs this thing, these entities called cells in which to exist, okay? They're the smallest units that have all the characteristics of life, and of course, as we said in cell division, a cell comes from a cell. Not answering the question about where the first cells came from, but that is still, that is a fascinating question, but that's more in scope with what I talk about in bio too. Cells stay small because they want to stay efficient, right? Cells remain microscopic because they want to maximize, where's my pen? Anyway, they want to maximize the amount of surface area. Now, when I say to maximize the amount of surface area, in your head, visualize what I'm talking about. If I'm, you know, convert the more technical term surface area to something in a cell. The surface area is the membrane of the cell. So when we calculated the other day that this was, what, 6 square millimeters? What we said was that hypothetical cell has a total of 6 square millimeters of membrane surrounding it. Now, as I mentioned in class, cells are rarely rectangular. Some are closely, which you'll see when we get together in lab, but for the most part, cells have blobular types of shapes, okay? But if we could theoretically shape a cell into a nice cube for ease of measurement, we would find, like I said, the one on the left-hand side had 6 square millimeters, where the one on the right-hand side had 24 square millimeters of surface area, membrane. So right away, when you look at that, you think, well, obviously the cell on the right has more membrane, and I told you, and very important in our next chapter, chapter 5, cells have to move things across the membrane, yeah? Just like you do. You don't move things across the membrane, but you move things across from the outside of your body to the inside, from the inside to the outside, right? Food comes in and waste products go out and all that sort of stuff. Same thing happens in a cell. All of those materials, all those waste products and all that food and all that other stuff that the cell does to be a cell, to be alive, happens in the volume of the cell, inside the cell, right? So what we're saying, and if we calculate the volume of this one, it was 1 cubic millimeter. And the other one was 8. I'm trying to do this with my mouse, and it's really weird. I'm left-handed, but I use the mouse with my right hand. So anyway, you simplify that. You simplify that to a 3-to-1. Okay, sorry, that's not coming out as a 3-to-1. So anyway, then we looked at class and we said, all right, which cell is most efficient? Which cell has more membrane around it in order to move those things in and out of its volume between the smaller cell and the larger cell? And it's the smaller cell, right? Because it has a 6-to-1 surface area to volume ratio. For every cubic millimeter of, that's right, for every cubic millimeter of volume, for every cubic millimeter, that cell has 6 square millimeters of membrane, where the cell on the other side, for every cubic millimeter, only has 3. So again, it's kind of like driving around here in Florida on the interstate. The one on the left is I-4 with 8 lanes across. My gosh, I remember in California when I lived out there that the freeways, both sets of lanes were 16 lanes across. It was amazing. I used to think to myself, how does some poor critter trying to cross the freeway? It's like Frogger from hell, right? Yeah, exactly. You would never be able to get across there if you were some critter. All right, so, and then the other one would be if they took those 8 lanes and shrunk them down, like because of an accident or road work, down to 2 lanes. You know what happens, right? It jams up, and that's the same thing that's going to happen in a cell. And now, without really getting too much into it, organisms and cells do things so they're efficient. And what I mean by that is not whether they efficiently, do something like we do at work, but they're efficient at doing cellular things, at metabolizing, producing waste products, producing cellular products. And they increase their efficiency by having enough surface area to bring materials across, raw materials that they need, as well as to get rid of waste products and cellular products that they make, right, which we'll talk about later, okay? So that's why when you walk around the mall, you don't see a big single-celled thing. You don't see a big single-celled paramecium hanging out at the Sparrow having a piece of pizza, right? So let's look at the different types of cells in a very, very broad category. Oh, good. Very good. Yeah, those, I feel so bad for the turtles because, when they get on the road, it's like sometimes they get stuck up against the curb and it's just they can't get over the hump and they just seem to, their brains just have a hard time trying to figure out. All right. These types of cells, these are bacterial cells. This is actually a TEM, transmission electron microscope shot of Bacillus coagulans, which is a bacteria. Now, these types of cells, these bacterial cells, are known as prokaryotes. There we go. And prokaryotes, if you remember from our first chapter, from one of our first classes, keep forgetting my video's not on and I'm pointing and everything, so you can't see me doing that. But if you remember, there were 2 domains that were prokaryotes. There was the domain called bacteria, and then there was these critters. Still prokaryotes, but in a different domain. All right, now there's I know there's a whole nother group of cells we're going to talk about in a second with plants and animals, but we're going to stick with the prokaryotes for a minute. One of the things that you notice when you look at a prokaryotic cell, and this is a very relative term, so please take it that way, is these organisms are relatively simple. I used to have a professor in college that hated when we used those terms, simple and advanced, because he used to say he was from Kentucky, and he'd say, they do so well with so little. Like if we were looking at a sponge as an animal, and we said, wow, there's not much, this is kind of a simple animal. He would say, yeah, but it's really good at being a sponge, so it's a very complex sponge. All right, so even though we look at this thing and we say, wow, it's small, half a micron, right, doesn't have a whole lot of parts, so it looks like a relatively simple critter. But again, remember, just because it's not complex in structure means that it's entirely simple, and maybe they would take offense if we called them that. Anyway, one of the things that you might notice about this organism, especially if you remember some of your biology, is there does not seem, in all of the labeling there, does not seem to be something called a nucleus, is there? Now, there is a nucleoid, but there's no nucleus. And you might remember from some of your bio courses that that's where the genetic material is, right? That's where the DNA is found. And all living things have to have DNA. All living things have DNA that is made up in these molecules that we call chromosomes. How many chromosomes do we have? How many chromosomes do human beings have, typically? Anybody know? A lot. There you go. Not a whole lot, actually, Jordan. For example, Some plants, I think it's watermelon, has nearly 1,000 chromosomes. Brandon says 23. He's right. We have 23 sets of chromosomes. More than 10 is a good guess, yes. We have 23 pairs of chromosomes. So a total of 46. Now, the reason why we say we have 23 pairs is you had a pair of parents, didn't you? Each of those gave you a set of chromosomes. You have the same number of chromosomes as your parents, so the sperm and the egg that your parents created must have half the number of chromosomes. Yeah, just do the math. So anyway, when those two half amounts of chromosomes get together, then the total number of 46 chromosomes is set back in motion again. Yeah, it's amazing. It is amazing when sometimes we look at the little changes that can occur. Yeah, but now remember now with a lot of, especially in the kinds of cells like plants and animals, a lot of the chromosome is actually not read or used. So all of us have genetic mutations in us, but there's just what we call silence. The mutations that I always tell folks, it's like if the junk mail coming to your house had misspelled your name, would you care? Not really. So if you had a piece of your DNA that was silent, that wasn't being used, that was mutated, no effect, no big deal. Yeah, good stuff. All right, but does this critter have a chromosome? Yeah, it's got one chromosome, that bacterial chromosome, that purple thing in the picture on the left. That's actually, if we unwound it, one loop, one connected circular loop. That's not circular. One connected circular loop of DNA. Really? How did you acquire these mutations, Jordan? So we take that loop of DNA, we fold it back and fold it back and fold it back, and then we shove it inside of that cell. DNA can be a pretty good sized molecule, so it's got to be folded around. Look at this word right here, though, and notice what it's pointing to. It's not pointing to a thing, is it? It's pointing to the chromosome. It's actually bracketed around the chromosome. You see, these kinds of organisms that are prokaryotes, they do not have a nucleus. And the reason why I focus on that is because it helps you remember when you look at the word. Pro means before, well, it has a lot of meanings, but in this context it means before. And kary, K-A-R-Y, is a scientific word we use for nucleus. So literally prokaryotes, those two domains, bacteria and the archaeans, are prokaryotes. They do not have a nucleus. Now, the before might throw you off, but the reason why that works in this context is these were the first critters on the planet. So before organisms had cells with nuclei, there were organisms that had, there was single-celled organisms that did not have a nucleus, but did have a chromosome, okay? That's very important difference between prokaryotes and eukaryotes. Prokaryotes, again, no nucleus and one chromosome. The rest of us out there, we have a nucleus and we have several chromosomes. All right, now let's look at some of the other structures there. A ribosome, we'll talk about what ribosomes do in a little bit more detail when we talk about the other types of cells. But the ribosomes are where this happens. I'm typing it. Right? I don't know how I got the little stick tongue out thing. It was supposed to be equals, and that's totally misspelled. But anyway, proteins are synthesized at the ribosomes, okay? Proteins are synthesized there. Now, let me ask you a question based on what we just talked about. So what molecules are being arranged in a ribosome? I know I said proteins, but what monomers do the ribosomes put together to make those proteins? Good little review for the exam, right? What are the monomers that make up proteins? Ribose is the sugar, Seth. Ribose is the sugar that you find in DNA and RNA. So that's the nucleic acid group. So think about the protein groups again. There were 4 levels of protein structure, and the primary level involved, I called it the recipe. The recipe of what? Not lipids. Lipids are fats, waxes, oils. You guys are bouncing all around the answer. Four groups of organic molecules: carbs, lipids, nucleic acids, and yes, amino acids, right? Amino acids make up proteins. So what's going on in the ribosome is that dehydration synthesis that I talked about with you guys, a couple weeks ago, and it's probably longer than that considering, with the hurricane. But that is going on right there in the ribosome. So ribosomes are where those amino acids are coming together in that dehydration synthesis, forming those peptide bonds. Remember, when we link amino acids, we have a name for it. We call it a peptide bond. Now, what's the difference between a peptide bond and a covalent bond? Does anybody know? If I ask you that on the exam, I love compare and contrast questions. So what if I said something on the exam like, compare and contrast peptide and covalent bonds? What would you tell me? Compare and contrast questions have two parts. How are they similar? How are they different? So if I asked you to compare those two, how would they be similar and how would they be different? Same kind of bond though, right? Isn't it, Seth? Same kind of bond. It's still a covalent bond, is it not? There you go. And the difference would be that it's only found in proteins or connections of amino acids. Perfect. Thank you. It's important to be able to associate these terms, because on an exam, I might ask you about a polypeptide. And you got to remember what that is. That's just another word for a protein, folks. Polypeptide means many peptide bonds, which by definition, protein, right? So we talked about the chromosomes. Plasma membrane, we'll talk a lot more about that in chapter 5. Obviously, that is the separation from the inside of the organism to the outside. Bacterial organisms, many of them, or I should say prokaryotic, many times have a cell wall around them. Now, we're going to learn about a cell wall in plant cells. Your cells, you animals out there, us animals, y'all don't have cell walls. Now, if you think, what do you mean cell walls? When we look under the microscope next week and we look at the onion cells, you're going to see a dark, semi-dark line around the outside of the cell. That's a cell wall. And cell walls, if you remember back from our discussion in carbohydrates, are made of cellulose. All right, so cell walls in plant cells, we discussed the fact that is what allows the plant to stand up against the force of gravity, right? That cellulose wall, it's a scaffolding. The cell wall in bacterial cells is a little bit different. They're not building large multicellular things like trees, but it does do a lot of the other purposes of the cell wall. It protects the cell, and it prevents the cell from what we say lysing, from bursting, for reasons that we'll talk about in chapter 5 too. Oh, the capsule's kind of neat. Some of them have a capsule. Sometimes it's a slimy coating. They use that capsule to, in some cases, to hide from your immune system. Yeah? Very nefarious life. Remember, your immune system is a series. Oh, this is a good review too. Your immune system is a series of cells that protects you against disease. How do your immune system cells, things like antibodies that we talked about, how do they know when they've come in contact with a disease organism, a pathogen, a bacteria? How do they know that? They don't know it in their head. How do they know that? Shape. Very good. Thank you, Brandon. Yeah, it's shape. They fit together, right? So these organisms have this, these immune system cells have these organisms to recognize these organisms by their shape. The capsule in many bacterial cells becomes thick, and guess what? It makes them essentially blind to your immune system. So now what your immune system is actually feeling is these lumps and bumps and these things that are sticking out of the membrane of the bacterial cell, and it feels those lumps and bumps, and it goes, those lumps and bumps aren't part of my cells. That's a different lumps and bumps. This must be a foreign cell. We should attack it and kill it. But here's what the bacterial cell says, is like, whoop, can't see me. Close my eyes, I've disappeared, right? I put a capsule over me, and now you can't see me. So the immune system cell comes along, is like, oh, I don't see anything, right? They hide from us. Bacteria, out to get us. The little fimbriae that you see there, which looks like little hair-like things, and I'm going to remind you maybe more than once, but certainly at least this time, only mammals have hair, so nothing else has hair. So it may look like hair, but it's not. Yeah, absolutely they do. Yeah, because it allows them to continue to replicate that shape so they can attack those pathogenic organisms like that. The fimbriae are used for a variety of things. One thing that the organism that causes strep throat, that organism, how it attaches to your tonsils, how it sticks there by fimbriae. Those fimbriae can act like little Velcro-ish type things, and they stick to host cells. There are also flagella, maybe one, maybe two, and they use those for motility, use those for moving around, for swimming around. This is what we call a typical rod-shaped bacteria. Bacteria come in three basic shapes: rod, round, okay, and spiral. The one that causes syphilis, the organism that causes syphilis, is a spiral-shaped bacteria. All right, so those are prokaryotic cells. Where are we coming away with this? Relatively simple. Put that word simple in quotes, but still, all the characteristics of life, right? No nucleus, right? They have a nucleoid region, one chromosome, one chromosome. Let's take a look at your typical animal cell. A lot more complex, right? Sorry to all you prokaryotes out there that are listening to me, but yes, this cell is a lot more complex. There's a lot more parts to it. There's a lot more stuff in the inside of the cell, which we call organelles. All them little things that are labeled, Golgi apparatus. It's a cool word. Got to be named after somebody. It does, kind of. It's got to be named after an Italian person who probably did work on that. Yeah, the reason why it looks like that, Jordan, is the way the artist has drawn it, as if you've taken the cell and you cut it open, like split it in half sort of thing. So they slit the nucleus and then cut the rest of it open. But yeah, it does kind of look that way. Now, we're going to go through each one of these organelles and talk about them, but I first want to kind of just go with a sort of an overview of what we're looking at. First of all, notice right away this critter has, or this cell, excuse me, could be a critter, has a nucleus. Now, prokaryote meant before, okay, and EU in science means 4, excuse me, and EU means true. All right, so these organisms are not prokaryotes, they are, I can spell it right, eukaryotes. They have a nucleus. Animals, plants, fungi, protozoans, all eukaryotes. The only prokaryotes are the bacteria, those two domains, the bacteria domain and the archaea domain. If you remember when we studied the domains, and hopefully you are studying the domains, there was one domain that was called eukarya. 3 domains: bacteria, archaea, eukarya. The bacteria and archaea are prokaryotes. The eukarya are all eukaryotes. They all have a nucleus. And they all have multiple chromosomes, right? We talked about us having 23 pairs. I mentioned to you that, watermelon had upwards of, I think, close to 1,000 chromosomes. All of those chromosomes, very important information, by the way, all of those important bits of information are stored inside of the nucleus. They're kept inside. Your DNA, your actual genetic code that we talk about, never leaves the nucleus. It's protected inside of there. It's double membrane, double bagged, really, because there's the big plasma membrane on the outside, right? There's that one. That's the cell membrane. And then you'll notice there's also, they call it a nuclear envelope, but it's a nuclear membrane. So we got a double membrane protection of that. The nucleus. You know what? Let me get to a bigger picture of the nucleus before I go any further, because I got another picture to show you. But notice that there are components called the cytoskeleton. We'll talk about those later, but cells don't have a skeleton like us, but we're using this word skeleton to refer to structures that help the cell maintain some rigidity. But these cytoskeleton structures have many uses beyond just forming a scaffolding for the cell, and we'll talk about those as we get to it. Peroxisomes, very bizarre organelles, a weird organelle to have inside of your cell, and you'll see what I mean in just a minute. Mitochondria, ooh, really fascinating story about these guys. They look like little beans. They actually kind of look like that rod-shaped bacterium that we talked about, just in the previous slide. They kind of look like that yellow one in the previous slide that we looked at. More on that in just a second. Jordan's asking, do they all have double membranes? Every membrane you see, everything has a membrane. All of those organelles have membranes. The cell has a membrane, plasma. The nuclear envelope is a membrane. All membranes are double membranes. All right? All membranes are double, in other words, they're double layers. So all cell membranes, like the plasma membrane, if you looked at it on an electron microscope, is actually 2 layers side by side. 2 layers of membrane. So it's actually, you think back what I said about the nucleus being double bagged, it's quadruple bagged, okay? We'll talk more about the double membrane when we actually get into membrane structure, but yes, absolutely, Jordan. But this is a good time to mention how important membranes are for the cell. Membranes allow the cell to do different things in different areas. And I mentioned the peroxisome just a minute ago. The membrane of the peroxisome allows that organelle, which when I explain to you what it is and what it does, it allows it to remain inside of the cell. And I'll get to that in just a second. Some eukaryotic cells have a flagellum. Sometimes they have more than one. You might remember this word sometimes, the cilia. Cilia and flagella are essentially equal in structure. The only difference between them is with cilia, there are usually many, right? Organisms like Paramecium have lots of cilia, and flagellum, usually there are few. Sometimes 1, 2. There's a few critters out there that might, I think about a half dozen, but they usually have much fewer flagellum. And flagellum also tend to be larger. You can oftentimes, well, not often, but if you get the light just right, hopefully, I'm going to bring in some pond water Tuesday, man, so you guys can just engorge yourself on looking at the world in pond water. But if you get the light just right, you can sometimes see it bounce off the flagella. Cilia are a little bit more difficult to see, but the only difference is size and quantity. Oh, yeah, if you get it just right, and you'll just see a flicker of that flagella. And that's why I like to use the pond water, because the critters are alive. Centrosomes, we'll talk about that. Centrosomes, if you look at them, what they're pointing to are these two, to me, I always say they look like 2 beer cans side by side. They look like one beer can standing straight up and another one laying down next to it. They're cylindrical objects. We'll talk more about what they do and their function a little bit later on. I'll talk about the endoplasmic reticulum, but I'm not going to say this word over and over again. My tongue would trip if I had to say, so I'm going to call it ER, because it sounds cool, right? We learned in class today. We learned about ER. Emergency room? No, endoplasmic reticulum. And there's a rough endoplasmic reticulum and a smooth, and it's a very complicated reason why one's called rough and one called smooth. Under a microscope, the rough one looks rough, the smooth one looks smooth. Now, I'll tell you why in just a minute, why the rough one looks rough, but I love that kind of stuff. You know, it's like scientists sometimes are sometimes kind to us. Let's not give it some fancy name for rough, let's just call it rough ER. Call it smooth ER. Now, look at the word endoplasmic reticulum. I'm a big word nerd. I like looking at roots of words and trying to figure them out. You know what endo means, right? E-N-D-O. What does endo mean? Inside. Okay. There you go. Inside. You got it. Plasmic refers to many years ago, and I was telling some folks I was looking through an old 1970s biology book many years ago when they couldn't really tell what was inside of a cell because of the microscopes and stuff. They saw a liquid filling is maybe the best way to think about it. All of these things that we know are there now that you see in the picture, we didn't have quite the knowledge about that back then. So they called that liquid filling, this was back in the 1600s, I think I was reading, or 1700s. Well, anyway, they called that stuff protoplasm. Protoplasm. Sounds like a cool name to name your kid or something, right? 1839, so I was way off in the years. A Bohemian physiologist named Purgy introduced the term protoplasm for that liquid filling, protoplasm. Now, we have another word for it. We call it cytoplasm. And that's basically the same thing. It's the liquid filling. And there's another word you sometimes see that's called cytosol. All of those basically are referring to the liquid parts inside of the cell. See, all of these organelles that you see, they're floating in this liquid cytoplasm, waterish, not waterish, full of water cytoplasm. Now, if you remember our discussion about enzymes and substrates getting together and randomly bumping into one another and all that thing I did in class, where I ran around and talked to you about the probability is what really controls those things coming together. Being that water is such a great solvent, right? One of the characteristics of water we talked about was the fact that it can dissolve most anything. All of those chemicals, those enzymes and substrates and all that other stuff, all that other inner workings of the cell is floating in this liquid protoplasm, cytoplasm, cytosol, nice, and it's mixing. It's a nice liquid medium for everything to mix. I say it's the social lubricant of cell organelles. It allows them to move around and to come in contact with one another, right? It mixes them together in a nice homogeneous mixture. Now, back to this plasmic part of endoplasmic. Endo means inside, and the plasmic is referring to this stuff is inside the cytoplasm. Well, it's inside the cell, so it's inside the cytoplasm. Now, Sean, reticulum. What does reticulum mean? I call on you because you're a snake dude, and that's a word that we sometimes hear a lot when we refer to snakes. There's even a reticulated python. Recall what reticulum or reticulation means. Reticulation. It's a cool word. When I was first in the Navy, having grown up in West Texas, no, not quite. When I grown up in West Texas, when I was first in the Navy, it was very much getting used to my sea legs when we went out to sea. Ship moving back and forth. So when I tried to walk a straight line down through the passageways, I wasn't very good at it. So I would walk back and forth. Yes, what kind of pattern, Emily? What happened to Seth? Okay, yeah, you get the right idea. A reticulated pattern is a back and forth, back and forth. Snakes move by reticulating. I walked down that passageway of that ship, reticulating, moving back and forth, okay? So the endoplasmic reticulum does, if you look at that picture, the stuff in the blue, and again, this is just what they did is they just cut it open so you can see inside. Because I want you to understand that not only does this stuff wind back and forth in the cytoplasm, it's a tube. That's what it is, right? Because you can see the part where they haven't cut, where I just drew the arrow. This is a series of passageways or tubes. So if you were to make just a swag. You guys know what a swag is? In science, sometimes you make a swag. In math, you might make a swag. What's a swag? It's an acronym. Stands for something. Nobody? Scientific, wild ass guess. Right? Have no idea. It's kind of like a hypothesis in a way. So if you were to make a swag, right, by looking at the structure of the rough ER and the smooth ER, right, what would you say the purpose was? What would you say that you think goes on with the ER? Yeah, they reticulate and move sideways. They reticulate off at an angle. When I was in West Texas, you could always, sidewinder tracks in the sand are always like this. They go up at an angle. Because that's, if you don't know, sidewinders do move exactly that way. They move at a 45-degree angle. Or not 45, they move at an angle. Nobody's got a guess for me what those little tube-like structures do? What if I tore away the walls from your house and we looked in a bunch of those pipes that run through the wall? What would you think those pipe conduit things would do? Well, what do those pipes do? I was thinking, this must be a trick question. Yeah, they move things. There you go. They move water. They move things. They carry liquid. That's what the endoplasmic reticulum does. It transports things. Now we'll talk a little bit more about what it transports in just a moment. But yes, see, that's why I want you to always do that exercise. When you look at these things and you're studying and reading about them, think about looking at it going, okay, I can see, yes, it looks like a series of pipes. So it's a transport mechanism. Now look at the Golgi apparatus for just a second. Notice it's a different color. They're really not this color, obviously. But notice the Golgi apparatus looks a lot like the ER, doesn't it? Now it has a little different function, but it still has a series of canals and winding ways by which things can be moved, all right? So we'll talk more about what they move in just a second, but that's just kind of looking at it at a very gross overview, right? A couple of cells that we're going to become familiar with. Oh, sorry. I forgot to go on to this one. Very quick. Animal cell on the left. Animal cell there. Plant cell, typical plant cell, animal cell, plant cell. Give me a big difference between the two. Give me one big difference between the two. If you have your notes in front of you, you can flash back and forth, but I'm not gonna. Oh yes, cell wall shape, perfect. Perfect, 1-2 punch, yes. The plant cell looks like, yes, it does have a cell wall. You see that brown around? That is the cell wall made of cellulose. You notice that a plant cell has a relatively regular shape. When you look at it under a microscope, you're going to see these are Elodia cells. They have a relatively regular shape to them, don't they? Not exactly a rectangle, but that way. Where animal cells, on the other hand, they're kind of like the shape of the proteins we were looking at, right? They're amorphic in shape. They're more globular in shape. All right, so that's one, the cell wall and the shape. Now, there's a couple of other differences. Can you pick them out for me? Think about what plants do that animals cannot. Chloroplast. Thank you, Victoria. See the chloroplast there? Yes, that's what they do. Chloroplasts photosynthesize. Do we have chloroplasts, us animal cells? No, we do not. No chloroplasts. We don't photosynthesize. Excellent, Caitlin. You're serving it up for me. Last difference between the two. Look at that big thing. Caitlin says that she notices that there's a big central vacuole in a plant cell. See that? Now, what is that vacuole? And it's a sac. It's a storage sac. It can store things. It's like saying, what is the purpose of a storage unit? To store things. That's all. It's not a living room. It's not your garage, right? It's the place to store things. It's a double membrane sac that plants store things. Now, here's a couple things they store. One of the things that plants store, like poison ivy, they store whatever gives you the itchy in their vacuole, right? So when you brush up against them, they deposit a little bit on your skin to remind you to not do that anymore. Remember the thing with plants, how do they protect themselves against predators? Do plants have predators? Yes, a herbivore is a predator to a plant. So how do they can't run. Maybe I can make it so you don't want to eat me, right? Maybe I can make it that once you take one bite of me and your tongue froths and it burns and it irritates you, you leave it alone. Exactly, right? Roses, same. Yeah, if you want to come and mess with me, you got all these thorns. I can't run, but I can protect myself, right? Now, another very important thing about plant cells and their central vacuole. And I'm going to go back again, just like I've done before, and just kind of start off with things you know, whether you've taken a science class ever or not. If you're thirsty, an animal, you and I, we can go get water, right? If a plant is thirsty, can it go get water? Can it move itself to go on down to the old watering hole to get something to drink? Of course not. They get it through their roots, don't they? Yeah, just like Jordan says, they suck up water through their roots, and that's how they get it. Now, if a plant was sitting in an area, a tree, and it was a drought, water was drying out, the roots were extending down, and water was just getting scarce and scarce, would it be beneficial to that tree if it knew that just a mere mile and a half away there's a big old oasis of water? Is that going to help the tree at all? It might help the elephant underneath the tree, right? Not going to help the tree. So between plant and animal cells, which cells do you think would have provisions to store water more? Now, both cells store water, but which one do you think would have a provision to store more water, the plant or the animal cell? Of course, right? Because they can't go get it. They got to store it. Now, we always talk about in science, form and function, form and function. What's it look like? How's it work? If you understand the connection of form and function, it helps you understand the overall concepts. A big central vacuole storage sac that might store stuff that makes you itch when you touch poison ivy, it's one of its most important jobs is to store water. Tons and tons, well, not tons, lots of water. Sorry, being a little bit hyperbole there. Lots of water. I would say the camel, because eventually the camel can go somewhere, right? Yeah. So not being able to, form and function, not being able to go to the water means that you're going to have something in you that allows you to store lots of water. Now, is that necessary in an animal cell? Yeah, we store water, but do we need to have something like that? Do we need to worry about those times when, man, there's water, but I just can't get to it? I mean, I understand there could be, but not untypically. Animals are going to be able to walk over to the watering hole, aren't they? Now, water is so important in storing inside of these cells that picture I showed you earlier, the Elodea cells. If you look at like this one, I'm going to put a box on. Look at this one right here. Couple of things are labeled. First of all, there's the cell wall. Those little green dots are the chloroplasts. In biology, there's kind of a rule of thumb. Things that are green photosynthesize. That's because chlorophyll, in order to photosynthesize, chlorophyll is green. It's a green pigment. So cells or things that are green typically photosynthesize. And that's not always the case. Like, Kermit the Frog, he's green, but he doesn't photosynthesize. But so, for example, this critter down below, Belonia ventricosa, by the way, the largest cell in the world, up to about 5 centimeters in diameter. So it's really pushing that surface area to volume ratio. It's photosynthetic. If you have aquariums, you might often see these algae. Well, that's actually in an aquarium. You can see the coral around it. But the chloroplast, the cytoplasm, all that sort of stuff. Now, if I could get you to look at that picture of the Elodea cells in 3 dimensions, and understand you're only seeing 2 dimensions here, understand it also has a thickness to it, you would see that all of those chloroplasts and all of the rest of the other organelles, they're all crammed up on the edges of the cell, right? As a matter of fact, the cell membrane, because plant cells have a cell wall and a membrane inside of it, the cell membrane you can't even really see because it's shoved up against the wall so tightly. Plant cells will store, when necessary, so much water that what happens is if you looked at the inside of the cell, my goodness, 99% of it will be that big central vacuole swollen with water. And all the other organelles will be crammed around the edges. Now, we'll do a lab later on where we look at how these plant cells change when they absorb and lose water. But let me get rid of my scribblings again. So can you see what I mean by that? So the green chloroplasts aren't in the middle of the cell. They're on the edges of the cell all around it. And that means that Elodea leaf is well hydrated. And I know that because I see a lot of clear spaces right here. Oh, thank you, Patty. I see a lot of clear spaces right there. Oh, yeah. We'll do it in lab so you can actually look at the microscope and watch this happen under the microscope. Tuesday's lab also involves you collecting human cheek cells off yourself. I'll show you how to do that if you haven't done it before. But look at that. It'd be really cool. These have been stained, which yours will be too. But what organelle is that? Of what, Jordan? What do you have slides on? Bring them in. Sure, we'll take a look at them. What is that organelle that I'm pointing to there? It's not going to be a vacuole because this is an animal cell. These are cheek cells. There you go. That's the nucleus. So you'll be able to see your nucleus under the microscope. That'd be cool. We all know about what stem cells are, right? The important and the issues with stem cells. Stem cells are cells that can be programmed to create any other type of cell. So once a cell has differentiated, once it's become a liver cell, even though all cells contain the same genes, your heart cell contains heart cell genes, your liver cell contains liver cell genes, and your liver cell contains heart cell genes. They all contain each other's genes. It's what genes get turned on. But once those genes get turned on, the other ones get turned off, you can't go back. Once you start becoming a liver cell, you have to become a liver cell. You can't say, oh, you know what? I don't want to be a liver cell anymore. I want to be a heart cell. Can't do that. Stem cells, you can then. Stem cells are at the beginning. They're waiting to be assigned their job. And in science or in medicine, you can imagine how beneficial that would be, right? Think about organ degeneration. Somebody's liver needs to be replaced. Now, we can do organ transplants, but there's many complications associated with that. Not the least of which is you're putting somebody else's liver in your body. Your body doesn't recognize that. So what they do is you could take your stem cells and grow another liver, activate the liver cells, or liver genes, grow another liver, and we wouldn't have to worry about the rejection. But then even be kept in track because they can grow. What do you mean grow out of control, Dorian? Grow out of control and then do what? Well, yeah, I don't know about them growing out of control, though. I mean, cells with uncontrolled growth are cancerous cells. That's what creates a tumor. Well, a lot of the controversy, of course, is how we harvest stem cells. But as far as the dangers in terms of operating them, turning them on and turning them off, I don't really see a danger. I mean, could somebody create some super monster genetic critter? Well, yeah, that could always happen. Scientific community really isn't that way, though. There's not a lot of mad scientists out there trying to destroy the world, to be honest with you, right? And if there are those folks that are a little bit on edge when it comes to, the work they're doing, the scientific community usually pretty well calls them out. Couldn't do that from stem cells. I don't think you could do that from stem cells. Or wings, right? Like I always wanted to be able to fly. All right, I talk more about stem cells in bio too, if that's something interesting. Yeah, become a spider, absolutely. Everybody's afraid of you then. Now, we're going to talk in Chapter 5 a little bit more detail about the membrane of the cell. Now, this is going to refer to all membranes of the cell. All membranes of the cell, whether it's the plasma membrane or the membrane around any of the, that would be cool, yeah. Around any of the organelles are all made of the same basic structure, and it's made up of two basic things. One of those you recognize, I hope you do anyway. It's a molecule we talked about just the other day. This one right here. And I said that it was amphipathic. All right, what does that mean? I told you that molecule there was amphipathic or ambivalent. It's a phospholipid, that's right, but the characteristic of being amphipathic, it's both. There you go, Seth. It's hydrophilic and hydrophobic. All right, now, I said if you took a bunch of those and you threw them into a glass of water, they're going to make a bubble, right? With the hydrophilic heads, this part is the head, attracted to the water, and the hydrophobic tails repelled by the water. Just keep focusing on what we're looking at here, looking over your notes. Don't worry, Jordan. So when we put those phospholipids together in water, they make this bilayer. You notice there's two layers of phospholipids. There's a head facing to the inside of the cell where there would be water in the cytoplasm, and the head facing out, and the tails are in between. This is what we call the bilayer of the cell membrane. This is a bilayer. Remember I was saying it's like a double layer. So the nucleus is like quadruply layered. So it's got the plasma membrane, and then every other membrane of the cell is made of this. The other major component that's found in cell membranes are these blue things that they've labeled protein channel, the integral membrane protein, but in general, what we call embedded proteins. All membranes in a cell are made up of phospholipids. I don't know if I spelled that right, but I'm not going to go back. And embedded proteins. That's the two major components of all cell membranes: phospholipids and embedded proteins. Now, we'll talk a little bit more in the next chapter about what these glycoproteins are and glycolipids and some of the things that these protein channels do. And a little later on the semester, I'll talk a little bit about what the cytoskeleton, these connections of the cytoskeleton. But I do want you to notice that the cytoskeleton, which is important for shape and rigidity, is attached, noticed, to the membrane. Now, these are all proteins, whether they're curlicue proteins or lumpy proteins. Those are all proteins, okay? So that's every cell membrane. This one has well, actually, I kind of misled you there. The nuclear envelope actually has an inner and an outer membrane. So there's actually 4 layers there. So the nucleus, or the chromosomes, are separated by 2 layers of the plasma membrane, 4 layers of the nuclear membrane, nuclear envelope. So there's 6 layers of membrane that things have to pass through before they get to the chromosome. Obviously, the chromosomes are pretty important, aren't they? You know, it's kind of like having 4 or 5 locks to get into a vault. Why do you think the chromosomes are so well protected? I mean, why do you have to pass through 6 different layers before you can make your way into the nucleus where the chromosomes are? Why are they so protected that way? Yeah, you don't want to do that. Don't want to mess up your chromosomes. Messing up chromosomes is like poking the devil with a short stick. You might get something really messed up. That's right. They're very important. Absolutely. That would be an extra chromosome. Yeah, being trisomy 21. Chromatin right here. DNA is a relatively fragile molecule, okay? And that means it can break apart, and we really don't want that to do. So what we do when DNA is not being used, it's not being read, it's not turned on by the cell, the DNA is wrapped around a protein, all right? And the combination, that protein, by the way, are called histones. And in my picture right here, the things on the inside, the little purple balls, those are the histone proteins, and the blue line, that's the DNA. The DNA wraps around the histones. And there's a bunch of histones, and it wraps that all up. And what that does is it's like putting white peanuts in if you're sending glass in the mail. It protects it. It kind of cushions it. And the collection of the DNA and the protein, the histone, is collectively known as chromatin. That's where that word comes. So the chromatin has the chromosome word in it. It was actually called chromatin because when they did the early work at investigating the nucleus, when they stained it, it took on a color. Chroma means color. So it actually takes on a fulgurant stain, a bright red color, actually. Even though the chromatin and the chromosome are well protected, there has to be ways for things to get in and out of the nucleus, and there are little things called pore complexes. Now, if you look at the picture down below, you can see where the artist has drawn a pore complex. A pore complex is a bunch of proteins. All these little purple things are proteins. What they do is they swell, right? And when they swell, they close the hole, and they contract, and it opens the pore. So proteins swell. So when they swell, they get bigger. They push against each other. They close up the pore, and when they need to open, they unswell, deswell, whatever the other word is, and they open back up, and things can move through. They look cool. Don't they look like little rosettes, aren't they? In the picture you see there. The nucleus also needs to be kind of tough, right? A couple of comments ago, you said you don't want it to get messed up. So if we look with again a TEM, transmission electron microscope, we notice that the surface of the nuclear envelope, we notice that those membranes are made up of what we call a nuclear lamina. Now look at that nuclear lamina. Doesn't that look like some structural material you might be familiar with? What does that look like to you? If you had to compare that to some structural material that we use for making cloth. Yeah, it's woven together, isn't it? Yeah. A who? But whatever. The cloth, the vest. To me, I think fiberglass. Fiberglass is a bunch of glass strung together in resin, and what that does is it makes it strong, doesn't it? So that nuclear lamella looks like one of Cooper's toys. Cooper has a way of ripping through his toys, so we bought him one that's got this really rough cloth, and it's hard for him to tear apart. So yeah, there you go. So the nuclear lamella, you can see, is very form and function. What does it look like? Looks like what you guys are saying. What does it do? It does that, right? Think about that when you're doing your studying. Look at it and go, yeah, it makes sense what it does. Now, I'm just going to, where the red arrow is up here, I'm just going to point out something here so that when I go into subsequent slides, we can connect this. Notice that the endoplasmic reticulum, the rough, which has those little dots on it, we'll talk about it in a minute. That's why it looks rough, by the way. Notice it's connected to the nucleus. Notice that the membranes are connected. All of this is part of something bigger that we'll talk about called the endomembrane system. All right? Any questions on the nucleus? Control center of the cell, top dog, head cheese, big boss. Right? If you have the DNA, if you have the instructions, you're the boss. Makes sense. Okay, ribosomes. Let's take a look at those. Those little tiny, now, there were ribosomes in the bacterial cell. There are ribosomes in prokaryotic cell. There are ribosomes in the eukaryotic cell. What do the ribosomes do in the eukaryotic cell? What's their function? Not transfer information, Seth. It does have something to do with information, or it does have something to do with using information to create something. What do we create in ribosomes? What did the prokaryotic cells do with their ribosomes? They made proteins, right? That's where they linked amino acids. Same thing happens here. All right, ribosomes actually, when we look at them more closely, are made up of two units, a large subunit and a small subunit, right? That's what one looks like. You can see a little, oh, look at that, man. Look at the picture of the ribosome. Ribosome is a protein, right? And, oh, let me go back one slide. I forgot to mention the nucleolus right here. There's a dot inside the nucleus called the nucleolus. And in the nucleolus, this is made. I'm typing it. One of those types of RNA, ribosomal RNA. Ribosomal RNA tells the cell how to make a ribosome, right? And a ribosome is a protein. See all the curlicues that you see there. But again, it does the same thing. It makes proteins. The only difference here, though, is you notice that some of those ribosomes are attached to the endoplasmic reticulum. Right? Let me go back to the big picture of the animal cell a couple slides ago. You notice that we've got ribosomes attached to the ER. So now you know why it's rough under a microscope. Those little studded ribosomes make it look rough. That's why it's called rough ER. Smooth ER does not have the ribosomes. And then there are even some ribosomes that are free-floating, that are not attached to anything. So we say in a cell we have a combination of bound and unbound or free ribosomes. Regardless of whether they're bound or unbound, though, they do the same thing. The bound ones are simply bound to the ER. The unbound ones are simply not bound to the ER. That's the difference between the two. Now it's time to put two of those things together. So again, let's take a look at a piece of ER and kind of draw it from a cross section. ER is pipe, right? Rough ER is pipe. So here's the rough ER cross section. So I'm looking down the pipe. Sitting on top of it is a ribosome. Form and function, folks. What is it that ER, especially the rough ER, what is it that it transports? I mean, it transports several items, but one real obvious one. What is transported inside the endoplasmic reticulum? Again, take a look at the proximity, they're attached, of the ribosome and the endoplasmic reticulum. Think about what the ribosome does, and then think about what the ER does. So what do you think the purpose of the rough ER is to do? That's a horrible English sentence. Hello, good morning. There you go, Victoria. Thank you. It transports proteins. That's right, it transports proteins. Proteins get transported through the ER. And oh, by the way, as those proteins are being transported, guess what they're doing? They're folding and twisting and making alpha helices and beta pleated sheets and folding back on herself. And if it's a big protein, maybe more than one of them are getting together. Remember all that stuff we talked about? All that's happening while it's being transported through the ER. Exactly. Form and function, right? The ribosomes being attached to the ER must mean, or fairly well indicates, right? Let's speak the science terms, that the ribosomes produce the protein, which is then dumped inside of the ER and then transported throughout the cell. Now, we'll talk about where that protein ends up in just a moment. But again, take a look at that. It makes perfect sense, okay? Now, the smooth ER transports materials, but of course, it doesn't have ribosomes attached to it. So we'll get to that in just a second, too. Ribosomes. Oh my goodness, this guy. Mitochondria. I'm not going to say a whole lot about the mitochondria because we're going to talk a whole lot about this in the subsequent chapter when we talk about metabolism. But I do want to point out a couple of things. Number one, it's got membrane within membrane, so it's got an inner membrane, it's got an outer membrane. Each of those is doubled. And then it has an inside membrane which winds back and forth. We call it a cristae internal membrane. Cristae is a term to refer, it's kind of like reticulate, winding back and forth. Inside of the cristae, or inside of these winding back and forth, is called the matrix. Now, what goes on here? Respiration, right? That's what we'll talk about in subsequent chapters, respiration. Those little windy membranes on the inside are crucial for the process of respiration for reasons we'll talk about when we get there. But I want you to make a good mental note of how much cristae, wound up, wiggly membrane you could stick inside that little organelle. Lots and lots and lots of cristae membrane. Membrane is important for the process of respiration, okay? Now, I'm going to come back to this slide in a second, but I want to jump to the next organelle and then kind of make my way through this next series of concepts that I'm going to go with. So the next thing I'm going to look at is the chloroplast. Again, I'm not going to spend a lot of time on this. We don't have them, but if you do, you use them to photosynthesis. Right? Got to have chloroplast to photosynthesize. You got to have chlorophyll. Got to be green. All right, this is a chloroplast. Notice it also has an inner and an outer membrane. It also has some stuff inside, things called thylakoids and granum and stroma. Don't worry about those terms right now. We'll talk a lot more about those when we start discussing photosynthesis. But I want you to kind of look at the big picture here. So notice, first of all, the shape. It's kind of a bean shape, right? Notice this shape. It's kind of a bean shape, right? All right, so number one, shape similarity, both kind of bean shape. Mitochondria have their own DNA. As a matter of fact, it's called mitochondrial DNA. Right? They have their own DNA. It's interesting about it. It's all maternal. Penny, what slide? What do you mean, which slide? All the DNA that's in your mitochondria just came from your mother. So shape, they have DNA. Chloroplasts have DNA. Look at that right there. Now, this may not be hitting you the way it should, but no other organelles have DNA, only the nucleus. All right, so we got a bean sort of rod shape. We got DNA that's independent of the DNA that's in the nucleus. It's like they have their own DNA, and If you decided to go out and start running, let's say, you decide to train for a marathon, your mitochondria is responsible for converting energy in your body. Well, if you started engaging in that kind of metabolic reaction, you would need more mitochondria. And that's what happens in your muscle cells. Your muscle cells divide too, but inside your muscle cells, they'll start making more mitochondria, right? Makes sense. So now you load up that cell with mitochondria, you have a lot more of those energy converting structures in there. The way mitochondria, and the same thing happens in plants, if you gave them a lot of light, they would start producing more chloroplast. The way they divide is very similar to the way prokaryotes divide. All right, let me go back, show you a prokaryote again. We'll make this mental connection. There's the shape, right? Kind of a bean, kind of a rod shaped. They have DNA. Mitochondria, bean shaped, they have DNA. These things divide by a process called binary fission, which we'll look at later on, but essentially they just split in half, right? They just split in half and form a whole new entity. That's the same way chloroplasts do it. That's the same way the mitochondria do it. Now, these sorts of observations did not go unnoticed by scientists. They thought, well, why would these organelles be so different? Why would they have their own DNA? Why would they look like bacterial cells or prokaryotic cells? And why would they divide very similar to prokaryotic cells? And what I mean by divide, in a muscle cell, the muscle cell doesn't have to divide. The mitochondria can divide inside of it. So the muscle cell can divide, obviously, but inside each cell, the mitochondria can divide through that splitting in half process. So like I said, this wasn't wasted on the scientific community, so they came up with an idea of perhaps how these organelles came about. So if we go back in time, oh gosh, 2 billion years, somewhere around there, we know, or we believe that what occurred is some ancestor of eukaryotic cells developed this ability to engulf things. Cells engulf things, like those old really bad sci-fi movies where the blob is engulfing the town. Eukaryotic cells are very well known for engulfing. They engulf food, they engulf things. So what happened in this particular case about 2 billion years ago is we believe that some ancestor of this eukaryotic cell engulfed some oxygen-using non-photosynthetic prokaryote. Mitochondria use oxygen, right? The process of respiration needs oxygen. That's why you have to breathe it. Instead of digesting and eating this prokaryote, this oxygen-using non-photosynthetic, it entered into a symbiotic relationship with it. It did the same thing with a photosynthetic prokaryote, for those that photosynthesize, right? So plants engulfed, they had engulfing of the mitochondria and engulfing of the chloroplast. What I'm trying to show is that the chloroplast and the mitochondria were separate critters. We believe that the reason why they have their own DNA and the reason why they look like prokaryotes and the reason why they divide like prokaryotes is because 2 billion years ago, that's what they were. They were prokaryotes. They were engulfed by the cell and became a symbiotic relationship. What's a symbiotic relationship? Did we talk about this? I can't remember if I did this in my other class or Bio 2. What's an example or what's the definition of a symbiotic relationship? There you go. It's a relationship between two organisms. It can be two organisms benefiting from one another. It can be two organisms in competition. Predator-prey is a symbiotic relationship. They don't have to be pretty, but a prey is interacting with the predator when it's being preyed upon. And of course, the predator is interacting with the prey. So yeah, so we can have positive symbiotic relationships. This would be a positive one. For reasons that we'll talk about in respiration, when these cells engulfed the mitochondria, they became much more efficient. Like on steroids more efficient, folks. So kind of back to that why they stayed small, engulfing mitochondria made very efficient cells. And of course, the benefit of having chloroplasts is obvious, right? I don't got to go out and find food now. As long as the sun comes up, how much longer do we have of that? How much longer do we believe until the sun fizzles out? Is it tomorrow? A few thousand years? Anybody have an idea? Take a guess. It's not before the end of the semester, so. Yeah, it's about another 5 billion years. About another 5. The sun is about halfway through its life. It's about a 10 billion year sun. We got about another 5 billion years. So photosynthesis is still good for another 5 billion years. So I'd stick with it. All right, we'll go back a couple of more slides. Point out something that there we go. See that arrow right there, the endomembrane system that I mentioned to you? All right. So the membrane of the nucleus is attached to the membrane of the ER, so they're attached together. All right, so let's talk about that. The endomembrane system. The endomembrane system includes these components. It includes the nuclear envelope, the membrane, the endoplasmic reticulum, something that we call the Golgi apparatus, which I'll talk a little bit more in just a second about what the Golgi apparatus does, and a couple of other organelles, which we probably, I don't know, we may get to some of these today. Lysosomes, vacuoles, and it includes the outside membrane, the plasma membrane. The plasma membrane in this context is going to be the cell's membrane. The reason why it's the endomembrane system is they're all related to one another because they're all connected by these things called vesicles. You can see it in the picture there. Vesicles are the little circles, bubbles, if you will. Now remember, everything in a cell is membrane, okay? So that transport vesicle is a bilayer of phospholipids. They're all made of the same thing. The membrane of the nucleus, the membrane of the ER, the membrane of the Golgi apparatus, all that stuff is made of this. The plasma membrane are all made up of the same thing. They're all made up of phospholipids in a bilayer. They're all made up of embedded proteins. Now go back to what I think chapter 2 when we were doing hydrophobic and hydrophilic, and I gave you a little adage. I said chemists tell you all the time, like dissolves like. Hydrophilic dissolves hydrophilic. Hydrophobic dissolves hydrophobic. Similar principle here. If you're made of phospholipids in a bilayer, you can mix with other things that are made of that. So just as kind of a little precursor to what I'm going to talk about, look what's happening right there. See that vesicle? This vesicle right here has something inside. Whatever that purple thing is supposed to be, maybe a protein. And when it touches the plasma membrane, because they're made of the same thing, phospholipids, they can fuse. And the fusion of those membranes causes that purple thing on the inside to be spit out into the outside. So like, for example, in your mouth, you produce an enzyme known as amylase, which helps you digest amylose or starch. Remember, that was the official name for starch. Your salivary cells would produce this enzyme, export it. So the enzyme would be that little purple thing in our picture, export it out into your salivary gland ducts, which would eventually dump that into your mouth. So that would be an example of how we export a cellular product. All of that is connected, that endomembrane system. All of the membrane material is moving through these different organelles by the use of these transport vesicles. So the ER that we talked about earlier, right? The rough ER has the bound ribosomes, and they secrete these things that we call glycoproteins. It's a sugar. Glyco means sugar, like glycogen. So the bound ribosomes secrete these things called glycoproteins. It distributes the transport vesicles, and it provides membrane for the cell. Because if you're looking at it, it looks like all the membrane would eventually end out on the plasma membrane. See, all these things are pinching off. It would almost look like all of the membrane of the cell would eventually end up on the plasma membrane, but there are mechanisms that bring membrane back into the cell. So that's why it's an endomembrane system. It continually is recycled. So the rough ER is attached to the nuclear envelope, okay, and along those there are ribosomes. The nuclear envelope contains inside the nucleus the DNA. The DNA makes its way out to a ribosome, creates a protein. The protein is dumped into the rough ER and then transports and does all its folding through there. When it gets to the end of that rough ER, it then pinches off into a transport vesicle. And this works just like if you took a piece of dough and you squeezed it, right? Or a piece of Play-Doh. And squeeze it, bloop, the little end would fall off the end. That's exactly what happens. So if you'll be patient with my lack of artistic ability, if this is the protein that we want to move, As it gets to the end of the ER, this part starts pinching in. Looks like I made a really nasty mouth or nasty face fish or something. So it ends up doing like this until eventually it pinches off into a transport vesicle. Now that transport vesicle winds its way through the cell, and guess what happens when it touches the plasma membrane? Just like we saw in that previous picture. It blends into it, right? And whatever was inside, like amylase, now it's outside. So you were very right about being transport. The smooth ER, which is connected, has some slightly different purposes. It's where fats are synthesized. It also helps to begin the metabolism of your carbohydrates. It begins to break down those carbohydrates. A lot of the metabolism is going to occur in the mitochondria, but some of it starts here. You're going to find a lot of smooth ER in the liver, too. That's because it also is beneficial for detoxification. So, and your liver is basically that. That's why I could never understand why people ate it. My mother loved the liver. Whoa. I'm like, gosh, Mom, you're eating the storage of all the junk. And it didn't taste good anyway. Calcium ions are also stored in the smooth ER as well, by the way. Calcium ions are important for muscle contraction and things like that. All right. We've got a few more minutes. I want to cover just a couple of real quick organelles, some simple ones. Well, I'm sorry, relatively simple. Does anybody have any questions for me on this endomembrane system? Let's talk about this last player in the endomembrane system, the Golgi apparatus. Now, the Golgi apparatus is the cell's shipping dock, distribution area. I don't know what you would call it in a company. It's where if you produce something in a company, it's the place in the company where all that would come together and be put on trucks. The loading dock. Jeez Louise, Steve. So that's what the Golgi apparatus is. The Golgi apparatus receives transport vesicles. So these right here. These are bilayer membranes with a protein inside that was formed from pinching off of the ER. Right? So it receives those on the cis face. So you can see what happened. It blends into it. Then the protein is dumped inside, or the material, not just protein, is dumped inside of the Golgi apparatus. It could be protein if it came from the rough ER, but remember the smooth ER also produces lipids. So it makes its way through there. When it gets to the other end, it repeats the process. It produces another transport vesicle. That's the trans phase. That's the shipping phase. See, the Golgi apparatus's job is to sort materials, right? The manufacturing is the ER. The Golgi sorts the materials and basically puts the address label on them, where they're going to go. Is that little vesicle that's formed on the trans face, is that material, was it made to stay inside the cell? Well, then it's going to go inside the cell and do what it needs to do. Or does it need to be exported out of the cell? And if that's the case, let's say it's this one, it's going to wind its way out to the plasma membrane and do what we've talked about before. It's going to actually merge to it. Okay? So we got the ER manufacturing. We got the Golgi apparatus, which is responsible for disseminating. Now, I'll leave you with the last slide with a relatively interesting organelle known as the lysosome. Lysosome is a transport vesicle, just like all of those other things, but it's what is inside of it that makes it important. All right, it's kind of like the thing when I told you the central vacuole and the storage unit analogy. A lysosome is just like any other transport vesicle that was transporting proteins or lipids through the ER that we just looked at. But inside of this transport vesicle are special proteins. They're known as hydrolytic enzymes. They are enzymes that are very good at hydrolysis, splitting down polymers. Lysosomes do two basic things. They digest food. It's on the left-hand side. See that cell engulfing? There's the food particle right there. The cell engulfs the food and makes a food vacuole. Along comes the lysosome with these digestive enzymes, these hydrolytic enzymes. Since they're all made of the same membrane, it attaches, fuses, protein, digests the material on the inside. So it's cell stomach kind of thing. All right, one more, and then I'll let you go. Polymers parts of a cell can go bad without the cell going bad. And I always use the analogy like if the starter in your car went bad, you could go out and buy a new car, or much cheaper and more efficient, just buy a new starter. So when something goes bad or something reaches its maximum entropy and is no longer efficient, like a mitochondria will say, lysosomes have a kind of a nefarious task. It's called autophagy. Now, phage, P-H-A-G-O, is to eat. So autophagy are organisms. No problem, Seth. We're going to be done in just about a minute. So autophagy is self-eating. And what the lysosome does is it blends with, don't worry about a peroxisome, I'll talk about that later, but the lysosome merges with this mitochondria that no longer is working anymore and digests it and recycles it. All right, so it's got digestion of food, getting rid of old cell products. And I'll go ahead and end it there. I'll stop the recording. Of course, then this will be posted later on the website. If you have any questions, stick around. If not, then have a good week.